Welcome to my channel, The Science Funda. Well, here is Ratna Vishwas again in front of the camera and for the students, for their welfare, with another video from the lesson, from the board I believe, ICSC. The name of the lesson is Absorption by Roots. And I am very fond of plants. So this whole world rotates on the green plants. Absorption by roots. Now, welcome to the new students and the old subscribers. Hope till now you have seen my old videos and getting some benefit out of it. And please subscribe, like and forward my channel to all the children who go for last minute revision. The background has been told to you by your teachers and the classes where you are going. I will be rotating over the key points of this lesson. It's a very big lesson. So, absorption by roots. You have been doing this lesson from your junior classes, that roots. That what is there which helps the root to absorb water and transport this water across the entire plant, whether it is a herb, whether it is a shrub, or whether it is a big coconut tree. Herb, shrub, or a tree. Now, what is the underlying principle behind it? Or what should be the features of the root which will allow the root to do this function? That should be the first question in your mind. Now this root, for example, this root, I'm not drawing it in detail, I'm just telling you, this root is a very unique structure found in the plants. Okay, and now why do the plants need water? Why they need water, and why the plants need water and minerals? Another question. What will you write? You should see, again I tell you, keep your ICSC book open in front of you. Water is required for photosynthesis. Water is required for transpiration. Water is required for transportation. Transportation of what? Transportation of materials. If this is a green plant, as you can see, the water should be transported across the whole plant. Now, how will it be transported? Only when the medium or the solvent as water will be found in the external media. Another thing is it shows mechanical stiffness. That is, water provides the cells to they swell up. Due to turgidity, you will learn a new term here. Turgidity, they become turgid. Like when we say, for example, when you soak a seed in water, the seed tries to imbibe water. Imbibe, because here I have written here what are the key words in the starting part of the lesson: imbibition, diffusion, osmosis. So this water is required for photosynthesis, transpiration, transportation, and to give mechanical stiffness to the plant. Now, next question for you will be, what are the characteristic features found in the root for absorption of water? What will you write here? Your first point should be, the root should have wide surface area. Like if the surface area is wide, the water will be absorbed more efficiently. For this only we see the trees which are not watered entire year and they wait only for the rainy season to come. They don't die away because the roots go deep into the soil till the groundwater level is there and they keep that water. This root gives them anchorage. This wide surface area of the root can help them to absorb the water from the soil to the entire plant, and this upward movement of water, that is from the root, if this is the root of the plant, to the entire tree of the plant, if this is a tree. This whole water which is going up against the gravitational pull, in your words, is also known by one word called as ascent of sap, which is there in the end part of the lesson. Ascent of S-A-P, sap. Now, what is this sap? This sap is a salt-like thing which is found in the mineral, the nutrients, all other things which are soluble in the vacuole. Because vacuole is the largest important cell organelle in the plant which plays the role of osmoregulation. It regulates the water content of the plant. And with this vacuole comes the ascent of sap, which you will see how vacuole will help this sap to get diluted with water and the forces acting for the movement of water upward, which is there in the last part, capillary action of water, transpirational pull of the leaf and the root pressure, that is in the end. I am starting with the key words like I told you. And for the first teacher of the root, which you will write, it should have a large surface area. Second thing, it should have the roots hairs containing the cell sap at higher concentration. They try to move from the outward to the inner part, because water is more in the soil than inside. So from higher concentration, they will go to the root. And also these root hairs are extremely thin. They are thin cell and they show the feature of semi-permeability. And these certain terms, semi-permeability, turgidity, are all related with osmosis, which you will come to see here. So surface area of the root is of a lot of importance. Second is the root hairs contain cell sap of a higher concentration than that of the surrounding water. The root hairs are nothing what are the root hairs? The fine unicellular root hairs. They are the extensions of the epidermal cells of the shoot. They also contain vacuoles which are filled with the solution called as the cell sap, as I told you. Okay, now, and many salts are dissolved in the cell sap, making the concentration of it higher than the water. So that is why water has to be absorbed from the soil to the root to make it dilute and then travel upwards. Okay, also the root has thin cell walls which are semi-permeable in nature. So these are the three important features of root. Now the very good diagram, figure 4.2, is given in the ICSE book. Kindly consult that diagram and draw, if they ask you, draw a fine diagram of root hair to show the absorption of water. This should be the diagram. That it has the outer cell wall, the cell membrane, the epidermal cell, and the large vacuole with the cell sap. You have to draw this diagram. Now we come to the next heading. As I told you, here is written imbibition. Imbibition. What is imbibition? So you should now here keep a notebook in front of you and write the tabular differences between all the three words which are written here. First is imbibition. What will you write here? It's a special type of phenomena by which water or any other liquid is absorbed by the solid particles of a substance, whether that part is living or dead. For example, you must have seen in the rainy season the wooden doors, they absorb moisture. So we say the wood has imbibed water into it. Water has not diffused into it. Water has not gone through osmosis, but through imbibition. So that is known as imbibition. And the solid particles which imbibe water or any other liquid is called imbibants. And the liquid which is imbibed is called as the imbibate. IMBIBATE. The liquid which is getting absorbed is known as the imbibate. Okay. And usually the water-loving substances or the hydrophilic substances have a tendency to absorb water. Like a dry seed, when we keep in water, it will absorb and it will swell up. And next example is that of the wood. And how it happens? It happens due to the imbibition pressure. Like osmosis will be due to osmotic pressure, okay? Diffusion will also be due to diffusion pressure, and all three terms are different yet interrelated. So what is the imbibition pressure? The pressure developed by the imbibant when submerged in a pure imbibing liquid called as imbibition pressure. Now, what is the significance of imbibition? All three things have their own significance. What is the first significance you will write? It helps, for example, the seed to germinate by breaking the seed coat. Second, it helps in movement of water from cell to cell in the plant body. Third, it is very important in the initial stage of absorption of water by the root. And lastly, it helps in ascent of sap, upward movement, because cell to cell movement that will only take place when it will help in the upward movement of water. That is for imbibition. Now, what will you write for diffusion? Say for example, diffusion. I can show you an example here. Say I take one this agarbatti. Simple agarbatti I am telling you, right? Agarbatti. This is, if you light an agarbatti, the fragrance of this agarbatti diffuses. It diffuses because it is from the higher concentration to the lower concentration. Same example is given in your book. When you take potassium permanganate and dip it in water, you will see the crystals of potassium permanganate are getting mixed with water and the whole color becomes violet in color. That is diffusion. So how will you call, what will be the definition of diffusion? What will be the definition of diffusion? You should write, it is the free movement of molecules say gas, liquid or solid from a region of higher concentration to a region of lower concentration without use of energy. It is a passive process without use of energy, and also it is a slow process, and secondly it is independent of the living system, and thirdly it doesn't require any selective permeable membrane. That is diffusion. Now, and the diffusing particles in a medium exert a pressure which is directly proportional to the concentration, and this pressure is known as the diffusion pressure. More the number of particles, more will be the diffusion pressure. So higher the concentration, as I told you. Now, what is the significance of diffusion? First, it is a means of transport of gases in plants, say the aquatic plants, the aquatic animals also. CO2 diffuses out, oxygen diffuses in. That is diffusion. Then the transpiration of water from the stomata occurs by diffusion. Thirdly, it distributes the ions and the molecules into the protoplasm. So you should now be very clear with the difference of inhibition and diffusion, right, in a tabular form. Last and never the least, the most important and which is related to you is osmosis. Osmosis. Now, how will you define osmosis? For you can please open page number 40, 41 where they are giving you some experiments of osmosis, and for that, what have they shown? Give you the definition of osmosis. It is the movement of water molecules from a higher concentration to a lower concentration across a selectively semi-permeable membrane. Osmosis takes place spontaneously in response to a driving force. It is also a passive process, and it also doesn't require any energy. So this osmosis is defined as movement of water molecules from a region of higher concentration to lower concentration across a semi-permeable membrane. Now there are different types of membrane. First type of membrane you should keep in mind, impermeable. That is, it does not permit the passage of either a solvent or the solute. Impermeable. Okay. For example, the cutinized wall and the suberin wall and the subidized cell walls, they are impermeable. Second is the permeable membrane. That is, it allows the passage of both the solute as well as the solvent. That is the lignified cell walls or the cellulosic cell walls. Third is semi-permeable membrane, which is what it allows the free movement of the solvent only, but it doesn't allow the solute to move in, or very selected solute can go, but only the solvent will go in. That is selectively permeable membrane. And for this only, in your book they have given you the example of which you have to write and understand the experiment to demonstrate osmotic pressure, where they have given you the whisking bag and that figure 4.4 to demonstrate osmosis, showing thistle funnel containing sugar solution. The thistle funnel is containing sugar, the beaker is containing water, but the level of sugar will rise up. Why will it rise? It's given here because the level of water in the beaker in the experiment will drop slightly, while the one in the beaker in the control will remain unchanged. All these reasons are given here. I am not going deeper into the experiments, but the conclusions which you draw from this osmotic experiment are: the water of the beaker has passed through the cellophane paper, which is acting as a semi-permeable membrane. Example of semi-permeable membrane is cellophane paper. So if they ask you, give an experiment to show the role of cellophane paper, this example will be there with this label diagram, figure 4.4, page number 41, where the cellophane paper allows the water to enter in through the thistle funnel, which is containing sugar solution, and thus the level is high. Now, sugar from the thistle funnel does not pass into the beaker, but water passes from the beaker into the thistle funnel. Okay, so the thistle funnel is the cellophane paper is acting as a selectively permeable membrane, which allows water molecules to pass through it, not the sugar molecules. It has been highlighted here. Secondly, if the same experiment is done with a rubber sheet, then what do you think? It will be allowed? No water will go because it is impermeable. What about muslin cloth? The experiments are asking you. Muslin cloth has very big pores, so it will allow the solute also to move in, that is the sugar in and out, and the water also too. You will not get exact result of the experiment. Okay, so this is what I told you about osmosis experiments over here. And what are the terms which you have to keep in mind here with osmosis are two important terms which is written here: endoosmosis, exoosmosis. Endo and exo. Now what is endoosmosis? What are the types of osmosis? Endoosmosis and exoosmosis. Endoosmosis, it is a inward movement when the water moves inside. Endo means inside the system, and it takes place across a selectively permeable membrane. Endoosmosis occurs across a selectively permeable membrane. It occurs in a hypotonic solution, and what is hypotonic I will tell you, and it makes the cell deplasmolized. The cell will be in a normal turgid condition. I have written the word turgid and flaccid. So these are the terms which you should know here. So endoosmosis is the inward movement of water across the selectively permeable membrane. It happens in a hypotonic solution. It makes the cell deplasmolized. That is endoosmosis. What about exoosmosis? Exoosmosis is the outward movement of water. It takes place in a hypertonic solution, and it makes the cell plasmolized. The figures of these different types of stages of the cell are given on figure in your book. It's given on figure number 4.8, that is page 44, where they have shown you the different types of solution: isotonic, which I will come now, hypertonic, hypotonic, and if they will ask you to draw a well-labeled diagram of these different types of solution in regard to a plant cell and in regard to an animal cell. Okay, before you go into this, you should know from where does these three terms come: isotonic, hypotonic, hypertonic. From where do they come? These three terms are derived from tonicity. The word I write here, tonicity. Now what is this word tonicity? What is tonicity? Very important question for you people. It is the relative concentration. It is the relative concentration of the solutions which determines the direction of diffusion or osmosis. I am telling you the relative concentration of the solution, which will determine the direction of whether osmosis will go in or out, diffusion higher to lower or lower to higher. And based on this tonicity, solutions are divided into three types: isotonic, hypotonic, and hypertonic. Isotonic means where the osmotic pressure is same on either side of the selectively permeable membrane. No gain of water, no loss of water. The cell will be in equilibrium. Isotonic. What is hypertonic? When the cell will have, the outside media will have high osmotic pressure. What is hypotonic? When the solution will have low osmotic pressure. The osmotic pressure is the key word here. So you should know the definition of osmotic pressure. Now, osmotic pressure is the minimum pressure that must be exerted to prevent the passage of the pure solvent into the solution when two are separated by a selectively permeable membrane. Or in very simple terms, osmotic pressure is of a solution is the measure of the its tendency to take water in. Osmotic pressure, in simple terms, is the tendency how much water can that system take in. That is osmotic pressure. We got three pressures we will read here, and also a very good diagram is also given in your book, figure on page number 45, where they have given you the figure of osmotic pressure, turgor pressure, wall pressure, which I will come later on. So this is osmotic pressure. So based on osmotic pressure will be the all these three types of solutions. Okay. Now this what is the significance of osmosis? This was in brief about osmosis. What is the significance? Like diffusion told, imbibition told. Now what about osmosis? What does it help into? First is it helps in the maintenance of cell turgidity. Now what is turgidity? Cell turgidity. Cell turgidity. What is cell turgidity? Turgidity means when the water moves into the plant cell, the vacuole gets bigger and bigger, pushing the cell membrane against the cell wall. This increases the pressure within the cell, making it turgid. The pressure exerted by the cell wall stops too much water to enter into the cell and prevents the lysis. So there are two kinds of pressure which are generated by a turgid cell, and we can call them as the turgor pressure or the cell pressure, wall pressure. So turgidity comes from the vacuoles, and the turgor pressure, and that gives rise to turgor pressure. Turgor pressure is from the protoplasm towards the cell wall, remember, by the protoplasm towards the cell wall, and wall pressure is reverse, from the cell wall towards the protoplasm. Diagram, that figure which I told you, which you will keep in mind, turgor pressure and wall pressure, on page number 45 ICSE book, a plant cell showing osmotic pressure, turgor pressure and wall pressure all together. Very important diagram. You have to write this and keep it in your mind. Okay, so that is the significance of osmosis is it helps in maintenance of cell turgidity. Second, it plays a very important role in stomatal movement of the plant, okay, during transpiration. Third, it helps in the movement of liquids across a biological membrane, and also mainly I have written here roots. Absorption of water by the roots is mainly through osmosis only. It is mainly osmosis, and through which I had told you the upward movement takes place, which is known by the word ascent of sap, which is later on. So this is the significance of osmosis. Now, another word which I have written here is, it is there in your lesson. You can now come on page number what? You can come on page number 44, where they are giving you the word turgidity and flaccidity. So turgidity is when the cell reaches a state where it cannot accommodate any more water, it is fully swelled up, distended, it is turgid, and this condition is turgidity. That is, the cell undertakes water by endosmosis and it is fully in a turgid condition. That is, it is in a deplasmolized state, which I will tell you now. And what about and what is turgor pressure? Is related to turgidity. The pressure of the cell contents on the cell wall is called turgor pressure. That is, by the protoplasm on the cell wall is called turgor pressure. Okay. Now, when the cell is turgid, it is somewhat in a balanced state. No more water is entering in or coming out. It's the turgor pressure which counterbalance the wall pressure. Both are vice versa of each other. The turgor pressure and the wall pressure are opposite to each other, and therefore there is no further absorption of water, even though the concentration of the solute inside the cell may be greater than that of the outside cell. Why there is no absorption of water? Because two forces are balancing each other, turgor pressure and the wall pressure, and this happens in a turgid cell. Now, turgid cell and turgidity and deplasmolysis are related. Opposite of turgidity, one word is there called as flaccidity. So, for example, again on your page number 45, when a living cell such as a cell of a leaf of an aquatic plant is placed in fresh water, it remains in a fully distended condition, fresh form, okay, but it's the plasma membrane remains in close contact with the cell wall and pressed against it, just like a rubber bladder of the football. I'm reading that paragraph to you. It pushes against the leather casing. If this plant cell is now kept in salt solution, they are telling you. First was plain water. Now the plant cell is kept in a salt solution, 5% salt solution. It will be seen that it loses its distended appearance and the cytoplasm will shrink. First feature of plasmolysis is shrinkage of the cytoplasm. That is the key word here. The cytoplasm shrinks in, and the plasma membrane will withdraw from the cell wall. This shrinkage from the cell wall is called as plasmolysis, and the cells in this state are said to be flaccid, which I have written here, flaccid. So turgidity and deplasmolysis, flaccidity and plasmolysis are directly related. Okay, so flaccidity is the reverse of turgidity, and got it. And what is plasmolysis? The shrinkage from the cell wall is called plasmolysis, and the cells in this state are said to be flaccid. Though flaccid and turgid are opposite to each other, but they are both related to each other also. And however, a plasmolysed cell is returned to water, then the dead protoplasm will again swell up, and again it will become deplasmolysed. So plasmolysis and deplasmolysis are also interrelated. So you have to keep all these things in mind for reading the lesson. So these are the keywords of the lesson. Now on page number 45, they have defined you the term turgidity, plasmolysis, flaccidity. I have also told you which you should write. We started with what is the root, what is the features of the root, told you. Then we started with imbibition, features of imbibition, significance of imbibition, diffusion, significance. Then osmosis. What is osmosis? What are the two types of osmosis? Endo-osmosis, exo-osmosis. Also told you the role of selectively permeable membrane, different types of membrane. That is, here you will write only cellophane as an example. Why rubber sheet is not a membrane, cannot be acting as a membrane, and why not a muslin cloth as per your ICSE book. Then we came to the term of tonicity. Three different terms: isotonic, hypertonic and hypotonic. How they are interrelated, and what is tonicity? Which cell organelle in the plant plays the role? It is the vacuole, because vacuole contains the cell sac, and it is here the relative concentration of water changes, and for this only the tonicity changes. After this we came to the term turgidity and flaccidity. Just now revised it for you. So these are the key words of the lesson. Now in your book, page number 46, students, some applications of turgidity are given, which you should also keep in mind. Turgidity provides rigidity to soft tissues, that is such as leaves. First point. Okay, for example, wilting of leaves you have seen is noticed when the plant is exposed to hot afternoon sun. But that same leaf, with the transpiration rate is reduced in the evening and little water is added, it again comes back to its normal condition. That is turgidity. Okay, then second is turgor. This turgidity helps in development of turgor pressure. As so this turgor pressure helps to push through the hard surface, such as the mushrooms. For example, mushrooms, when they grow into the different types of mushrooms are there that you eat. They grow on the tree trunk, they grow on the rocks and all, on the anywhere they grow. So how do they grow? Due to this turgor pressure. And the roots of certain trees, they have been seen to crack the walls of the concrete floor of an adjoining building. If you keep observing, you have seen all this. The plants are growing on cement, rocks and all, because of this turgor pressure. Because against turgor pressure is playing a very, very important role. Now, two applications. Third, they have also given you an application is turgor in the root cells builds up root pressure, and also this root pressure is responsible for what? For this ascent of sap, which you will read at the end. Root pressure, and the experiment is given here in your book. Kindly see that experiment of root pressure which is given there. Now, if they ask you that what is the significance of plasmolysis, a hot question sometimes they ask you, what will you write here? What can be your answer here? Plasmolysis, the significance of plasmolysis can be, it is used to kill the bacteria of contaminated food. If the food is contaminated and bacteria is there, the bacteria which are growing on that, you can add a large amount of salt to it, so that all the bacteria will kill. So this adding of salt is making the cell plasmolysed. So plasmolysis application. Second is, it is also used to kill the weeds which are growing in the garden. That is the application of plasmolysis. Now another application of turgidity, if you have seen the touch me not plant, Mimosa pudica, what happens? The turgor movements, the water content in the plant helps the plant to either to open up. Your touch is the main stimuli is there. Touch me not, it is called that plant. Either it will be in the drooping form or it will be in the open form. That is due to the turgor pressure. Also, there is one very specific tissue there in the plant. It's given in your book on page number 47, pulvinus. What is pulvinus? Pulvinus is a special tissue found in this Mimosa pudica plant, which develops turgor pressure in it, and with this the plant opens, droops, and or comes back to its original condition. Now we turn to the page, come to the last part of the lesson, where they have given you the diagram, figure 4.13, how the water goes across the plant, which I told you in the starting. These are this was the background of the lesson. Now the last part of the lesson is this tree and the water moving up the tree. Now how? For this, you all should know the structure of root. If we take a root and cut a transverse section of the root, some layers are there arranged in sequence. The first outer layer is known as the epidermis. Epidermis has the fine unicellular root hair. After the epidermis, I am verbally telling you, you can make a flow chart. First is the root hair, then is the epidermis, third is the cortex. After cortex is the endodermis. As we go deep, after the endodermis is the vascular bundle, and in the vascular bundle is the xylem and the phloem. This xylem helps in transportation of water, and this transportation of water is a unidirectional condition, up, But transport of food is bidirectional, which is done by phloem, and that is called by the word translocation. When the food is transported by phloem, we call it as translocation. And when water is transported by the xylem, we call it as plane transportation. And there are special cells in the xylem which helps in doing it. What are those two cells? The xylem tracheids and the xylem vessel cells. These tracheids and vessels are the special kind of xylem cells which are having pits and perforations in them, and they form a connecting channel-like structure inside the plant, like a pipe. One pipe connected to another pipe, like a long pipe, and the water goes up, up, up. These are the tracheids and the vessels. This yellow-like thing you can take as the perforations or the pits through which the water is going. The water goes from one pit to the other pit. One cell has the lower concentration and the another cell inhibition, and inhibition is also playing a role here for making the water move up. And this water, when it moves up across the vacuole, is known as ascent of sap. And for this, the root pressure, the amount of pressure built up in the root is very important. How much pressure is built up in the root. So that was the last part of the lesson. I told you the layers. The layers are given in your book, figure 4.13. Kindly draw that diagram and learn it and keep. And after this comes the very important term is there in your book, guttation. Guttation, guttation. Now what is that guttation? If they ask you, it's a term related with transpiration, but it is different. So how will you write this word? In certain plants like tomato, grass, banana, etc., when the root pressure is very high and the force of water is very high, what happens? How much water? And what does transpiration do? Transpiration helps the water content. It gives a cooling effect. The water regulation of the plant, it helps a cooling effect. Guttation will not give you the cooling effect. What will happen here? All the water will come out from the stem or from the ends of the leaf veins. Okay, and this water appears as tiny drops along the margins of the leaves, which you will see in the next lesson in the early morning and all. And this loss of excess water is called as guttation. So this is called as guttation. Okay. So I told you about this guttation. I told you about all the important points of the lesson. And in the end, there are certain features here which they have told you to keep in mind. What is ascent of sap? Ascent of sap, as I just now told you, is the upward movement of water. And what factors on which it is dependent on? Root pressure builds up sufficient force to push the sap in the xylem vessels up to a certain height. That is root pressure. Next is capillary movement. Capillary movement is narrow the diameter of the xylem vessels, higher the water will go up, and narrower the diameter, greater will be the height of the water and the exerting force or the capillary force. Third is the transpirational pull. That is, higher the rate of transpiration, higher will be the more the water lost by the leaf by the process of transpiration, more water molecules will be pulled up due to the tendency of the water molecules to remain joined together. We call, when same water molecules are joined together, we call them as cohesion, adhesion cohesion theory. And when the water molecules are joined with the walls of the cell, we call it adhesion. The adhesion cohesion forces also play a role in ascent of sap. First is root pressure, second is capillary movement, third is transpirational pull, and fourth you can write adhesion cohesion forces. And apart from this, your book has given you certain important experiments which they can ask you to write and draw. First experiment to show that the absorption and conduction of water in the plants. Root is the main structure here. You have to draw this experiment. Figure 4.15. Next is to show that water is conducted by the xylem. They are taking the plant and putting it in eosine solution and cutting a transverse section. They will see the vascular bundle has taken a pink stain. That is this part of the experiment. Third experiment is that they have shown you to see that conduction of water takes place through xylem. The experiment to the water is conducted through xylem. This experiment, and the fourth one is to show that food from leaves is conducted downward through the phloem in the stem, where the the girdling experiment here and the girdling experiment on both the experiment 3 and 4 are very important for you to write and study. So the whole lesson actually is now divided into certain, you can say, the quick revision of the lesson in 2 minutes. Absorption of water and minerals, what features of the root it has for absorbing water, why water is important for the plant. Then came what is imbibition, what is diffusion, what is osmosis. Also came the significance of all the three, but with the differences also of all the three. And then came what is osmosis, what is osmotic pressure, what is tonicity, three types of tonicity, hypo, iso, hyper. Then came flaccidity, rigidity. Then came deplasmolysis, plasmolysis with their significance. And in the end comes ascent of sap and the factors which cause ascent of sap. I think I gave you a quick revision of the key words of the lesson, absorption of water by the roots. So thank you from my side, and I hope I could make you the terms clear, and rest the diagrams and all you have to draw, revise and study. And our subject is learned by reading, writing and speaking. Thank you dear students. Keep watching and liking my channel.